Thank you, Megan. So next we'll hear from Dr. Omar Abdel-Wahab, who is a former Damon Runyon Clinical Investigator. He's an associate member at Memorial Sloan Kettering Cancer Center. Omar?
Well, good morning, everyone. Thank you all so much for the opportunity to speak and for being here. So as Young just mentioned, I'm a medical oncologist working at Memorial Sloan Kettering Cancer Center, and I focus on blood cancers, and I'm also a physician scientist that focus on the molecular genetics of blood cancers. And so in 2012, I was fortunate to receive a Damon Runyon Clinical Investigator Award, and I want to just share with you all one story that's come out of that work that we're really excited about that hopefully directly illustrates the impact that this research has had on patients. So as many of you likely know, for decades, the way that we've diagnosed, classified, and treated cancers has been based on the anatomic site within the body that cancers arise, as well as how cancers look under the microscope. And so, for example, a patient with lung cancer or kidney cancer or glioblastoma were all diagnosed and treated separately based on where their cancers arose. However, many of you likely are aware that over the last 2 decades or so, it's been understood that maybe trying to genetically understand cancers or understand their biologic characteristics might be a better way to develop therapies for patients and linking specific biologic or genetic characteristics with specific treatments. And this has been led to a lot of progress over the last 20 years or so for cancers. At the same time, there have been challenges. So for example, many of our so-called targetable mutations are rare with any single form of cancer. Secondly, is that even when this process works, resistance to therapy almost always seems to occur eventually. And so to illustrate this, you heard a lot about melanoma from Megan, and we didn't coordinate this, but this is just a nice example. This is a patient with melanoma skin cancer with a common mutation in melanoma called the BRAF mutation. And this is a PET scan, and you can see these dark spots within the abdomen. That's the metastatic melanoma. There is fortunately an FDA-approved inhibitor of the mutant BRAF, and within 2 weeks, the patient, you can see there, those black spots rapidly resolved. However, and unfortunately, as the patient continued therapy, they developed resistance just 14 weeks later, and you can see that by the reaccumulation of spots. Now, this BRAF is a signaling component within the cell and normally controls cell growth. And when it has this BRAF V600E mutation, it results in increased accumulation of cells and a cancer develops. Now, interestingly, this same mutation, this one mutation, is present in many diverse forms of cancer: melanoma, non-small cell lung cancer, thyroid cancer, et cetera, many common forms of cancer. And as I mentioned, there's this FDA-approved inhibitor called vemurafenib, or another drug called dabrafenib. But as I mentioned, I'm actually a blood cancer doctor, and when these stories were developing, we noticed that actually this mutation seems to be incredibly rare in our common forms of blood cancer, very different than solid tumors. However, there's one form of disease that I was seeing clinically and always really curious about. It's a rare form of blood cancer called histiocytosis. Many of you probably never heard of this. Many of the physicians in the audience probably never heard of this disease. But it's thought for years to have been a kind of inflammatory disorder, not even known to be a cancer. And as we were beginning to study the genetics of this disease, we found that nearly half these patients had this BRAF mutation, the same mutation seen in melanoma very commonly, colon cancer, et cetera, as I mentioned on a prior slide. And this disease is very unique. It's fascinating clinically. It can affect both children as well as adults, and it impacts almost any part of the body, as you can see from these clinical images. But the discovery of this mutation led us to think, well, could we target this with the drugs that are used, as I mentioned earlier, for melanoma and these other common cancers? And so we started a clinical trial, and it was just enormously successful. This is one of the first patients we treated. And on the left-hand side there, you see the MRI and PET scan from pre-treatment, and the white spots in the brain there are a disease within the brain that affected this patient. She couldn't even sign the consent form herself to initiate the trial. Her husband had to do it. But within 2 weeks of therapy, there was rapid resolution of her disease, as you can see in the brain there and the PET scan around the kidneys. Another patient, this is an image of a PET scan again, and you can see the disease in the bones in this individual around his knees, ankles, wrists, et cetera. And on treatment, those rapidly resolved. Just one last example. This is a patient, I'll never forget, actually nearly came to us blind, and you can see the disease behind the eyeballs there in the green arrows, and there was rapid resolution of the disease behind the eyes, restored vision, and has been in remission now for 2 years. So this has been a really recurrent observation and led to the first FDA-approved therapy now for this condition in 2017. But as I mentioned earlier, we knew that only half the patients with this disease had the mutation and could get this therapy. So we began to dig further, and we found that the other half didn't have mutations in BRAF, but had mutations that activate a protein just downstream there called MEK, or MEK. And here again, we had parallels to melanoma, where there was an FDA-approved MEK inhibitor called cobimetinib, and there are several others that were already approved for these unrelated forms of cancer. So we thought, okay, let's do another trial for these individuals using this drug. And the results, I think, were even more exciting than vemurafenib. One of the first patients treated, a PET scan here. You can see there the pre-treatment PET scan with the disease in the bones, and again, the resolution of those lesions. Just one more example here. This patient had disease throughout the central nervous system. It was very debilitated, but then actually really rapidly recovered their neurologic status within starting this therapy. And unlike the solid tumors, for reasons that we don't understand, these individuals, they don't develop resistance as far as we can tell now for 3 or 4 years to this therapy. There's something unique about this form of cancer that's been really interesting. So with that, I want to conclude and tell you that a number of genetic alterations are present across diverse forms of cancer and may serve as important therapeutic targets that we can treat these cancers actually similarly, even though they're quite unrelated clinically. And as an example, I showed you this one disease targeting BRAF and MEK has been really important for this rare form of blood cancer called histiocytosis. And so we now work to implement this so-called precision oncology approach, which is not a new concept at all, for many other forms of blood cancers and other mutations. And I want to stress again that support from the Damon Runyon Clinical Investigator Award really made this possible. And again, Lorraine and Young have really supported my career now for the 7 years since I met them. So thank you so much for your attention.